Yeah, so I'm here to close things out with talking about model validation. Much as you automatically get this after refinement. So when you're doing structures, you automatically get it after refinement when you're in the tutorial. And I like to start this out with some words of encouragement about validation can make a difference. So what you can see here is tracking clash score over time in the PDB. So this is a database level phenomenon tracked for sort of a crucial middle resolution of structures, 1.8 to 2.2, average clash score in the PDB. High numbers are bad, low numbers are good, and you can see that prior to clash score being introduced in MolProbity, you have sort of it's relatively high. Once MolProbity is introduced and people can see this, it starts improving. And once it becomes available to more people through the Phoenix GUI, it improves even more quickly until we reach what appears to be some sort of stable point that is about as good as we can do at the moment, which I think is also interesting. So this is at the database level, validation can make a difference. And also, I want to note that you personally can make a difference. So this tracks an epidemic of too many cis peptides deposited into the PDB. I'll get into exactly what those are in a few slides, but this is an extremely rare phenomenon. Way too many of them were being modeled, and one of our collaborators, Tristan Kroll, noticed this, wrote a paper, and everybody started paying attention. Partly people making tools that were more aware of this, and partly just people like you knowing that this was a problem and watching out for it. And we get this incredibly steep decline in this over-modeling problem back to sort of the base rate of how often we see this. So you personally can make this kind of difference this fast if you pay attention to your structures. And I want to make a few points about overall validation philosophy before we get into specifics. In particular, visualizations are more important than statistics, and local confirmations are more important than structure level averages. Whole model averages can hide both the interesting things and real problems just because you average out. You can hide a real problem behind everything else looking good. So you have to look at your structure and you have to look in detail. And also be aware that outlier thresholds are set statistically. These are, in fact, statistical outliers. So when you have enough residues, you expect statistically to see a few outliers, especially at interesting sites like active sites. So don't get rid of these. Cherish them. You found something cool. Maybe write about that too. And points about philosophy of intervention into structures. Refinement is great at details. Refinement is better at details than you're ever gonna be. But it is relatively bad at escaping the wrong local minimum. So when you are using validations to go into a structure and make changes, your first priority should be on finding the right local minimum for whatever the feature is, and then preserving any interesting details you find. So if you have some of those outliers that are real things that are interesting, make sure you keep them. And then just don't sweat the details. Get to the right local minimum, then put it back into refinement and see if it sticks, see if you're happy with the result. But don't worry about every last geometry detail until you let refinement take a shot at it. So I'm going to talk about a lot of validations. These are available in Phoenix through the validation GUI that we'll do a tutorial on a bit. They are also available on the MolProbity website if you want to use the web server and the way it presents information. You get basically the same thing. If you want a quick and dirty version of some of this tutorial information, if you go to the MolProbity website, there are more tutorials down here. I'll put some more links to these at the end. In particular, this guide to validation options. has links to relevant papers for each of the validations. So if you need to follow through into more specifics, this is a place where you can get some of them. And of course, this is all open source, and if you want to install and run MolProbity yourself, you can. All right, on to the meat. So we present a lot of validations, and I'm going to run through as many of these as I can. So for each of these, I am going to briefly describe the underlying idea of how the validation works. I'm going to show you how outliers in that validation are presented in King or our NGL viewer or Isolde, because it really helps to be able to look at these things. And I'll try to give a couple useful examples of things that might cause this outlier. Not at all comprehensive. The possibilities for mistakes are vast and wonderful and keep me busy. But ways to start thinking about what you might see. So, first of all atom contacts and clashes. This is sort of the signature validation of the Richardson lab. And for this is the thing for which it is most important that you add hydrogens first. You can do this with phoenix.reduce. You can do it through the MolProbity website. As we said, it happens automatically in most refinement. And once again, it is important because without the hydrogens, you are missing half of the atoms. So if I add all the hydrogens to this, you see how much denser this image becomes immediately. Hydrogens are responsible for at least one side of almost all the contacts in macromolecules. So to do our all-atom contacts, what we do is we take a 1/4 angstrom radius probe sphere and roll it over the surface of the van der Waals surface of each atom. And where that sphere touches or overlaps with another van der Waals surface, we mark that point. And we mark that point according to the kind of contact that it is. Note that this analysis does not do anything explicit about angle, which can be important for hydrogen bonding, but does just inherently deal with certain kinds of shielding effects. So hydrogen surfaces can shield heavy atom surfaces. So you can see that the surface for this oxygen actually disappears once it gets underneath the hydrogen. So there are some advantages to this technique. If you really need to know about angles, it doesn't really do that. So when you have favorable van der Waals packing, you get these nice green and blue dots that pack in close together. Where you have favorable hydrogen bonding, you get these light green pillows, and unfavorable steric overlaps called clashes get these hot pink violent spikes. There's a nice visual intuition built into this in that the more dots you have, or the larger the spikes or the larger the pill, the stronger the contact is. So that is, the strength of contact is built into the visual intuition here. So when you see clashes, what are they telling you? Well, clashes are usually an emphasis on other problems. Usually, if you have a real modeling error, it will have a clash associated with it because it's very hard to get the contacts all right if the other things aren't. One of the things this means is that when you have real outliers, they are much less likely to have clashes associated with them, and they're more likely to have things like hydrogen bonds or very close packing that holds an otherwise unfavorable conformation in that interesting position. In the case of outliers, clashes can also help imply directions for fix-ups. So in the example we have here, this is a rotamer outlier. It's a bad side chain. And what you can see is there are a bunch of clashes on this side. Well, that implies maybe you should push it in the other direction. And wouldn't you know, there's a little nubbin of unfulfilled density there. Well, those two pieces of information together make it pretty clear what you should try doing. And indeed, if you refit the side chain over in that direction, Now both of those things are fixed. You fulfill the density and you get rid of the clashes. So you can get directional information out of this. This example of a sidechain error is as good a segue as any into sidechain rotamers. So protein sidechain conformations are described by a series of chi torsions, chi1, chi2, chi3, sometimes chi4 as you go further out the sidechain. And rotamers are the statistically expected combinations of those chi values. For tetrahedral atom centers, like most carbons, this means staggered conformations, plus, trans, minus. If you have a planar atom center, the rotamer distributions are much more continuous and get named after a central angle value. So here you see rotamer distributions for isoleucine, chi1 on the x-axis, chi2 on the y-axis, and you can see that mostly this falls into those staggered plus, trans, minus combinations, and you get several very well-defined clusters that are rotamers, and you also get some combinations that are not statistically represented, that are really unusual things. Not necessarily impossible, but you better have a justification if one shows up. So if you're looking at these in King, we trace rotamer outliers over the side chain in gold. If you are looking in Isolde, which is available through Chimera X, you get these corkscrew spirals, which are color-coded by prior probability. The redder it gets, the less likely a conformation this is. Likely sidechain rotamer problems. It is relatively easy to fit certain kinds of sidechains backwards, especially these ones like valine and leucine and threonine that have a branch at the end. You can flip it around and fit the terminal atoms into the density about equally well at minimal expense to the branch point. But this ends up with a staggered conformation that you'll see in the tutorial. So this is not a real thing that sidechains want to do. This is pretty easy to fix with just a flip, but you got to watch out for it because it's actually a little hard to see from just the density. More complex cases, sidechains can get stuck in the wrong density. Sidechains can get stuck in density for all sorts of other features like other sidechains and ligands, and at lower resolution, it can even get stuck in the backbone. And you may have to fix an entire network of problems to fix the sidechains. So in the example we have here, the foundational error is this tyrosine is stuck in the wrong density. And if you get the tyrosine out of that wrong density and over where it belongs, which you can see down here, then there's space for the arginine to be in its proper place, and that gets the arginine out of the backbone density over here, and you can build something that actually looks like a helix again. So you have to fix this whole thing at once, which is the kind of thing that refinement is going to struggle with, and you have to apply some human intuition to get this into the right local minimum. So moving into the protein backbone and sort of a family of validations that help deal with that. Ramachandran and Kablam and Rama Z-score. So if you know only one validation, you've probably heard of Ramachandran analysis. This is the absolute classic. These phi and psi torsions describe local protein backbone conformations. And so we take those, and each residue's phi-psi pair gets converted into Cartesian coordinates and checked against contours of expected behavior. As Oleg indicated, one of the interesting things is that there are actually multiple distributions for Ramachandran space. Besides the general case, we also have lysine, which, because it lacks a side chain, is much more permissive. We have proline, which, because it comes back and attaches to its own backbone, is much more restrictive. And in fact, proline is so restrictive that we define a special case pre-proline for residues that are immediately before proline, because they are also noticeably restricted in their and the conformations they can take on. And isoleucine and valine, because they have a branch at the C beta atom, that interferes with the range of things the backbone can do. So they also get their own special distribution. Visualizations. In King, we draw this green bar centered at the C alphas. In Isolde, you get a ball at every C alpha that is color-coded by Ramachandran favorability, from green to favorable to red for unfavorable and outlier. Moving on to Kablam, and this is some of my very personal work. Kablam is meant to deal with lower resolution structures where the detail level is less compatible with what Ramachandran analysis is doing. And the basic idea is that at low resolution, the C alpha trace of the backbone is modeled better than the other backbone details. And many common modeling errors involve wrong peptide plane orientation between those C alpha positions. So what Kablam does is it uses the modeled C alpha trace geometry to predict likely peptide plane orientations, and where the modeled peptide plane orientation disagrees with the predicted one, we mark that discrepancy as a probable outlier. The root causes of both Kablam and Ramachandran outliers are basically misplaced carbonyl oxygens, especially as you get to lower resolution. Electron density for the carbonyl oxygen starts to disappear, and you can get that peptide plane fit in arbitrary orientations. And you get these low resolution density envelopes that allow multiple models to fit within them about equally well, without discriminating very much between things that are protein-like and things that are not protein-like. So Kablam and Ramachandran can help you make that distinction. Pavel already talked about the Ramachandran Z-score earlier, so I will cover this lightly. It is a way of looking at the overall Ramachandran distribution and seeing if it is realistic or seeing if it is possibly overfit. What I want to add to that is, rather than the whole distribution, pull out a couple of specific examples to help give you an intuition for why it's important to be careful about Ramachandran restraints. So here I've pulled out just one pair of residues for each of these cases. So here's something that's supposed to be an alpha helix, and we're looking at this residue pair here in the middle, where the peptide plane has been turned about 90 degrees out from the axis of the helix. The Ramachandran positions of the residues on either side of that are marked with these purple balls, which you might be able to figure out from these Ramachandran positions that they are supposed to be alpha helix, but you might not. They've gotten a little far from the actual ideal alpha helix position. The beta sheet example I have here is even worse. So you've got, instead of alternating, you've got 3 carbonyl oxygens all pointing in the same direction. So if we look at the residues on either side of this, our target is the beta region, but one of them ends up over here, and one of them ends up all the way over here, because you're moving about 180 degrees in Ramachandran space. There is no way that you can tell what these are supposed to be from their current position in Ramachandran space. So, and in fact, they're both within allowed contours. So my point is that current Ramachandran position does not necessarily predict correct Ramachandran position. So restraints don't solve errors. Restraints keep correct structure where it should be. So you have to fix things first before the restraints. All right, C beta deviations. This is kind of an unusual one. So because the C alpha is a tetrahedral center, the ideal C beta position is defined by backbone geometry. And we can calculate the ideal C beta position based on based on the average of these two torsions using backbone angles. And if we observe a C beta that is more than a quarter angstrom away from that ideal position, we mark that as an outlier. This results in one of our coolest visualizations, I think. So we draw this, for C beta deviation, we draw this magenta sphere. The center of the sphere is drawn at the ideal C beta position that we calculate. The edge of it is drawn tangent to the modeled position. So as your model gets further and further from the ideal position, the sphere gets larger and larger. An elegant visual intuition for how severe the outlier is. I wish we could do that well for everything. We also provide this bullseye kinimage that shows not only the distance, but also the direction of all C beta positions. The yellow ring here is the quarter angstrom outlier cutoff. So when you see one of these, you probably won't be surprised to know that the C beta that is positioned at the interface between the backbone and the side chain, if there's a C beta deviation outlier, it probably is a problem with the interface between the side chain and the backbone. Usually it means there's a misplaced side chain that is pulling on the backbone in a strange way. Especially those branched side chains fit backwards can result in a large C beta deviation. A special detail to watch out for is if you have a misnamed chirality. So if you have a D-alanine, which should be named DAL in your file, and it's named ALA for L-alanine instead, you'll get enormous C beta deviation because switching from L to D flips the sign on those torsions. So you get an enormous C beta deviation. Generally, Phoenix is good at bookkeeping, so you won't see that if you're completely within system. But if you're working between systems or importing old structures, keep an eye out for that. You will sometimes encounter mislabeled chirality swaps. Covalent bond geometry is next, and really I can't make this one exciting. You measure bond lengths and angles, you check them against the library of expected values. If they're too far from the expected value, it's an outlier. The thing that is interesting is that Phoenix uses a confirmation-dependent library as our targets for this. So that is based on local Ramachandran confirmation rather than being just one value per bond or angle regardless of context. For the markup, bond lengths are drawn as bond length outliers are drawn as springs, bond angle outliers are drawn as fans, and if you know a little bit of astronomy, these are intuitively color-coded. Things that are too far get red shifted, things that are too close get blue shifted, and the way the fans and springs are drawn gives you an intuition for the direction that this should be pushed. So, probable causes of geometry problems. Sometimes you will encounter systematic errors. So these tend to occur in programs with different libraries or different expectations for geometry than what Phoenix has. The example I have here is from a prediction by OmegaFold, which actually, thank you, Christopher. You did this for me the last time we had this workshop here. At that time, OmegaFold had presented systematically short peptide bonds, systematically so short that almost every peptide bond in the prediction is an outlier from Phoenix's perspective. So be aware of what you import. If you are entirely within one system, you probably won't see this sort of thing. But if you move from system to system, you may pick up some weird stuff. Usually a geometry minimization or re-refinement will just sort this out, but keep an eye on it because occasionally things can explode if the geometry is bad enough. Rather than that sort of chronic case, you can also have acute geometry outliers, of course. Localized outliers generally result from either having to put conformational strains somewhere and it goes into a vulnerable place in the structure, or from missing density. So in the case you have here, this is mostly a great looking structure, 1.3 angstrom resolution. Almost all of it looks like that until you get out to this loop and there's just no density at all. So everywhere else in this structure, you could downweight the geometry restraint. You could downweight the geometry restraints and rely just on the map or more heavily on the map. And that works great. And then you get out to this loop and that strategy fails and fails badly, and you get something very unphysical. To fix this, you may be able to manually apply more restraints to low data regions like this. You may be able to rebuild it from scratch. And people always ask, what should you model things like this? And my answer is that nobody who comes after you is going to know as much about your structure, your system as you do. So model everything that you can, because nobody else is going to be able to do it. But if this is the best you can do, and maybe it is, it's better to leave this out than to put in something that is blatantly non-physical, because proteins don't do this. It's better to leave this out if this is the best that you and refinement can manage. So cis peptides, I mentioned this earlier in the context of things you can really make a difference on. The peptide bond that joins adjacent amino acids has partial double bond character and does not rotate freely. It therefore takes on either a more or less planar trans configuration or a more or less planar cis configuration if my fingers here are the C alphas. It has a special relationship with proline, and about 5% of all proline residues are preceded by a cis peptide bond. So when we get to the markup, we color those in. You get this characteristic trapezoid shape when you get the cis configuration. We color that in with this nice sea green so that you can see where it is, but it's not offensive. Cis before anything else is extremely rare. 0.3% of non-proline residues are cis. So we mark this with this bright lime color so you can't ignore it. And occasionally you get peptide planes, again, this is supposed to be planar, that are twisted more than 30 degrees away from planar. That basically should never be happening. So we mark those yellow and please don't do that. This basic color scheme maintains through Coot, where you can also see these, and Isolde, mostly just changing what color the cis non-prolines are. Probable causes for cis peptides. The nasty thing about these is that when you the C alpha distance, this is shorter in a cis configuration than in the trans configuration. So if you have patchy density, if you have kind of little blobs of density, that shorter C alpha distance looks like it fits into that fragmented density better. A conformation this rare requires more than just a marginally better density fit to justify. Think about how rare this is. You need density that is proportionally that much more supportive of the unusual conformation. So unless you know something from the density or chemistry or homology or another source, and you might, model your residues in trans configuration by default. So we do offer some RNA validations. I've mostly been talking about proline, though things like contacts and geometry applies to everything. For time, I am just going to gloss over these. There are extra slides at the end, so if you download these off our website, you can take a look at those later. We offer validation for protein RNA backbone conformations, which are rotameric if you break them up the right way. And we offer validations for the ribose sugar has two pucker states, C2' endo and C3' endo. And we have a way of very accurately predicting which one of those it should be, regardless of how it's actually modeled. So if you're an RNA person, check the extras for details or get in contact with us. And we have some very exciting stuff coming on that's not quite ready. All right, with that time instead, I want to talk about UnDowser and water validation. And if you are here for sort of expert level stuff, perk up your ears now because this is the most difficult validation thing. So UnDowser is a tool for helping find incorrectly modeled waters. So if you look at this example, here's a water. It has enormous clashes with this side chain. What atom, so clearly this water is wrong to be here. What atom should this be instead? And anybody want to audience participate? What should be in this water position? That's fine. It should be the other part. It should be the other end of the side chain here. So UnDowser tries to provide advice for every clashing water. It tries to provide advice on what that might be instead. You can get this. The easiest place to get this is still on MolProbity. Run through, click on this UnDowser button, and you'll get an HTML chart. It is now available in Phoenix on the command line. It's phoenix.undowser_validation. By default, it will give you this HTML chart. You can also get JSON. I want to run through a few examples of waters in the wrong places to give you an idea of the kinds of things you might be looking for. So here's a water that should be an ion instead. So besides having a little bit of a clash here, you can see, and this is a stereo image, so you may be able to do the cross-eyes thing to get depth, which is really helpful on this one. This has an unusually strong density peak, much stronger than you'd expect for water, but one which would be consistent with a metal. It has octahedral contact geometry, which is the thing you'll be able to see better if you can get the stereo. Not only does it have 4 on each side, there's also a contact on bottom and a contact on top. Water doesn't do that, ions do. Less important, but still useful here, the contacts are all to partial negative polar groups, which is another good sign that maybe this is actually a positive ion. And in this case, it's probably a sodium. On sort of the far other end of things, here's a water that just shouldn't be. There's no density peak for this at all, which is the most persuasive thing. It has these clashes. Its contacts are to a mix of polar and non-polar things, which is unlikely for an ion. An ion would probably be mostly touching polar things that are coordinating with it. So all of that comes together to tell you this water probably doesn't really exist and should just be removed. Waters can also get stuck into the density for other things. Like here's 3 waters jammed into the density for a ligand. They clash extremely badly with each other, and waters that clash very tightly with each other, waters that are within covalent bonding distance of each other, basically, that's a strong indication that some kind of real other molecule is supposed to be there. Could be a ligand, could be the end of a side chain, things like that. You can also get waters that are part of an alternate conformation network. So here we've got a backbone that has an A alternate and a B alternate, and it's clashing quite badly with a couple of non-alternate waters. Well, without changing these water positions at all, because there really is density support for both of those, you can resolve the whole problem by just renaming the alternates so that this is an alternate A water and this is an alternate B oxygen, so they don't see each other anymore. That was just renaming. That didn't involve deleting anything or rebuilding anything. And you can also get waters that replace alternates. So here we have, this is a disulfide bridge. It has a bunch of those very close contacts, and they are contacts to non-terminal side chain atoms. They're contacts to atoms within the side chains. which is another indication that this is probably actually supposed to be more sidechain in an alternate conformation. To be fair, to be clear, waters can be real too. What you do actually get waters in structures. They can have density. So here's a water with a clear density peak. It's weaker than the macromolecules density, but that's fine. You actually kind of expect that for waters. It has some hydrogen bonding, and again, this is weak evidence, but it has contact with both partial positive and partial negative polar partners, which is the kind of thing that water does, and mostly not the kind of thing that ions do. The kinds of issues you run into with water are vast and fascinating, and this is just a sampler. So feel free to, when you download slides, feel free to follow these links to a paper and one of our Phoenix newsletter articles for more. If you are using this, an important note, since we haven't updated some of our rhetoric with the validation yet, but since then we found that the interaction distance for negative ions is generally too far to see a clash when a water replaces one. So clashes with partial positive polar partners probably indicate alternates or other unmodeled structure rather than indicating negative ions. So there's a little bit of not quite accurate advice in some of the validation feedback. Pavel and I are planning to work to develop some more water validation tools once I finish the exciting RNA stuff. That runs through all of the specific validations that I was going to talk about. So I should close this out by talking a little bit about our MolProbity score. The MolProbity score is a single score that combines several validations and then scales the result to look like a resolution. It combines clash score, it combines Ramachandran validation, and rotamer validation. And because it's scaled to look like a resolution, if your MolProbity score is better than your model's resolution, that's a good sign. If your MolProbity score is worse than your model's resolution, that means you probably have more work to do. But as I said at the start, a single statistic really cannot adequately explain a whole structure's quality. So don't rely on that, especially at low resolution, because the MolProbity score was tuned to work better at higher and mid resolutions. Hopefully, through this workshop, through these lectures, you now know enough to look at the other statistics, and you now know enough to look at your model and the markup in detail and get into the stuff that may be hiding behind that whole model average that the MolProbity score presents. Freshly added, based on the earlier conversation, when do you stop with this? Realistically, you do as much as you can. Ideally, you stop at the point when you and refinement can't make the structure better. You might think of this as convergence. It's basically when you can't make any more meaningful changes. It's important to remember that zero outliers is not the goal. Some outliers are truly justified. Some outliers are not justified, and you're just going to be stuck with them. And that is life in structural biology. And again, if you can't obtain a physically reasonable solution, and sometimes you're just not going to be able to because you don't have the data for it, consider deleting the region and leaving it out of your model because it's something that's unphysical, isn't real. I pointed you to these links on the MolProbity page earlier. For a quick version of the material I've covered here and links to our publications, there's the guide to validation options. I've deliberately glossed over the structure level statistics because I want you focusing on the details. But if you want the summary of what our whole model targets are, then follow this link to the summary table guide, also available on the MolProbity website, and you can get what those targets are. And again, if you download the slides, there's some more validations after this.